Good morning, good afternoon, and good evening to all of those on the Zoom today. Ugo, I really appreciate what you've done to organize this meeting and your perseverance to make sure that we gather this year. So I really appreciate that. So today, what I'm going to do is give you a snapshot of where we are with respect to the work we've been doing on muscle IGF-1. And as you know, or as many of you know, my lab has been really focused on how insulin-like growth factor benefits muscle in terms of health and prevention of disease. And for many of what we've been looking at two aspects of what IGF-1 does. First, it has an anabolic role in muscle, and that primarily works through the IGF receptor to promote protein synthesis, increase regenerative capacity, and prevent atrophy. But it also serves a metabolic role in muscle, and it does so through primarily through hybrid receptors that upon a binding of IGF-1 behave more like insulin receptors and help with glucose homeostasis and handling in muscle. So the working hypothesis that we've been governed by for the past several years is really saying that muscle IGF-1 can both directly and indirectly modulate both growth and glucose homeostasis. In order to study the necessity of muscle IGF-1 for these processes, we developed a deletion scheme, a mouse model where we can induce deletion of IGF-1 at any age. And this is just a simple crossbreeding of a fluxed IGF-1 cassette that was developed almost more than 20 years ago, crossbred with a doxycycline inducible skeletal muscle specific Cree. And then we've also bred in a Rosa reporter so that we can make sure of the specificity and extent of deletion. And what we know is from both DNA and RNA analysis that we can get very effective deletion of IGF-1 in muscle. And further, just looking at the reporter, the Rosa reporter, we can see that with doxycycline introduction to these mice, we can see that there is effective deletion within the quadriceps, but no deletion in the heart or the muscle. And this is just a snapshot of what we've been looking at in a recent publication by Yorgos Vasilakos, a postdoc in the lab. So in order to evaluate what's going on with muscle IGF-1, particularly in adult mice, where this balance between anabolic and metabolic actions becomes really more key, is that shown in the left-hand side of this slide is the scheme that we've introduced where we can add doxycycline to the water or food and then analyze any given number of weeks afterwards with respect to IGF-1 levels, body weight, muscle morphology, and a number of different anabolic and metabolic parameters. But shown here is the fact that we can see that with this deletion that we see about a 50% decrease in muscle IGF-1, which really tells us that muscle itself can make its own IGF-1, but the circulation also provides a source for IGF-1 in the muscle. And so we can never really fully delete IGF-1 from that tissue, given that it's a secreted factor and that the blood supply provides an additional circulating source of IGF-1. But further, what we can see down here is that the serum levels of IGF-1 are also diminished, telling us that muscle itself also provides a source of IGF-1 for the entire body. And so we just have to bear this in mind that in this particular model, and I think we're probably as close to the closest we can get to deleting IGF-1 from muscle is this profile we show here. So in addition to the fact that we have this lower IGF-1 levels in the tissue and the blood, we also saw in these animals where we deleted at 12 weeks of age and then looked at 16 weeks of age that we saw the the animals became fat. So we had an increase in percent of adiposity within these animals. We also began to see disruption of glucose handling, both in the fasted condition, where there was an inability to clear or reduce glucose after 16 hours of fasting. With respect to glucose clearance, we saw an impaired glucose clearance, where you can see that there's not only delay in the clearance, but also an inhibition of clearance acutely. Further, what we saw was when we exercised these mice in a run to exhaustion test and looked at glucose levels in the blood prior to and after exercise, we saw that unlike the wild type mice shown with the solid circles, where there was a reduction in glucose after an hour of exercise, the mid mice shown in these diamonds did not display the same reduction in glucose. And so in all of these features, we see that this is beginning to tell us that there is something going on with the reduction of IGF-1 with respect to glucose handling. In addition to looking at these glucose handling measures, we teamed up with Julia Ayala, who was at the time at Sanford Burnham Prebys in Orlando, to look at a number of more whole animal assays with respect to activity. And what he measured was the fact that the respiratory exchange ratio shown here was shifted to lower values indicative of the mice, the mid mice, using fat utilization as opposed to carbohydrates. Further, when we looked at food intake, there was a reduction in the food intake over time, and the mice were not very active. And so all of these tell us that, again, there's this reduction in activity, which would potentially lead to that increased adiposity. But in addition, there was a change in how much they were eating and what type of energy they were utilizing. To follow up with this idea of this the changes with respect to IGF-1 deletion, using RNA-seq of muscles from the mid mice versus the wild type mice, we found that there were a number of differences, both up and down regulated genes that were in a number of different categories. But in particular, what we saw was the fact that with reduction of IGF-1, shown here just in the expression level, we saw this selective drive for atrophy, shown with the upregulation of atrogenes. We also saw a shift in fatty acid and lipid metabolism, which was shifting again towards genes that were supporting the enhancement of fatty acid metabolism. And further, we saw that there was an impairment in glucose handling, in particular with genes that were important for helping to translocate GLUT4 upon insulin or any other activity monitor of GLUT4 translocation to enable glucose clearance through muscle. So with respect to all of this baseline characterization of these mice, what we're seeing is that there is both reduced muscle and circulating IGF-1 levels. There's diminished muscle mass. There's decreased bone mass, which I haven't shown you, but we have now accumulated data on that. There's increased fat mass, impaired glucose handling, and diminished activity. And I would argue that many of these features are really mimicking or hallmarks of both type 2 diabetes and aging. And so now, even though it's not an explicit model of aging or diabetes, we are certainly grappling now with this idea that reducing muscle IGF-1 really leads to some of these potential pathologies that we could use this mouse model now to enable us to see if we can therapeutically alter these trajectories of pathology. And so what I'm talking to you about today is a study that was just completed by Ray Spradlin, who has just defended his PhD and has moved to University of Illinois, Urbana-Champaign. My most recent picture is during COVID times, and so you can't see his smile, but I think under the mask he might be smiling. But in the second part of his dissertation, he asked the question of how well do mid-mice adapt to endurance exercise? And further, the subtext of that was, can exercise improve their health? And so what Ray did was design a training program that was initially based on lactate threshold limitations, so that the first phase of an exercise regimen was at a moderate speed that was just under the lactate threshold for these mice. And then after 4 weeks of time, he increased the speed of training to just maintain this training effect and continuously push the mice so that they would continue to improve their endurance. The whole mouse study was started at 16 weeks of age, where we induced deletion and waited 1 month. And then intervening times between prior to the start of training, in the middle of training, and at the end of training, we ran a run to exhaustion test to monitor how well these mice ran. Further, in addition to all of these whole animal activity parameters, we also looked at body composition, glucose clearance, and finally at the end, harvested tissues and looked at isolated muscle function. So in terms of IGF content, just to be sure that the deletion was effective, Ray looked at a number of different regulators of IGF-1 in terms of gene expression by qPCR, but also ran ELISA analysis of IGF-1 in both the muscle and in the serum. And what we saw is similar to our prior work with this model is that we had easily a 50% reduction in IGF content within the muscle. And further, we had a 30 to 40% decrease in the amount of circulating IGF-1 within these animals. Exercise itself did not alter the level of IGF-1 either in the mid-mice, which had the deletion, or their strain-matched control mice shown here. Further, in respect to all of the regulations that we saw, the absence of IGF-1 or the absence of the in muscle did not alter the training effect of a number of these different parameters with respect to binding proteins or myostatin or its superfamily member activin A. So taken together, we see that there is no overall effect of training on IGF-1 content within muscle. So if there is this change in fuel utilization within these animals, one thing we wanted to test was to determine whether or not there were aspects of the pathways used for fuel utilization differed within these animals. And just shown on the right-hand side is the one rate-limiting step for uptake of fatty acids into the mitochondria for beta oxidation is the protein CP1B. And in using qPCR and in our RNA-seq data, we saw that there was an upregulation of expression of CT1B in the mid-mice. And further, with exercise, we saw the control mice had an increase in expression of this gene. Further, when we looked at protein levels, we saw that there was this 7-fold increase in CT1B protein in the mid-mice, again, supporting the idea that these mice really are relying on fat utilization, on fatty acid metabolism, instead of carbohydrates. When we did the run to it, I keep saying we, it was really Ray who did this, but I'll keep on saying the royal we. The training speed is shown here in the red square to show you what that level was with respect to the run to exhaustion test. And this is a very classic run to exhaustion test that many have used for mice, in which every 3 minutes, the mice increase their speed over time. And then at the point at which they fail to run with encouragement, we consider that the time at which they stop running, and then we record that distance. So in this run to exhaustion test, what we saw initially was that there was no difference in the run to exhaustion distance run by any of the mice prior to their exercise training. And that's just shown here in the run to exhaustion test. As the training went on for 4 weeks, we saw that there was a significant improvement in distance run. And this was similar in both the control mice and the mid mice, and there was no difference in the sedentary mice compared to their training, to their pre-training status. But as we increased the intensity to 18 meters per minute for the following 4 weeks, what we saw was that there was a separation of the ability for the training effect to continue, where the control mice, shown in blue, increased their distance still further in terms of their ability to run, yet the mid mice failed to experience or show that improvement. And in fact, in many cases, the distance run actually decreased. Further, at 8 weeks of time being sedentary in the cage, we began to see the separation of mid mice from their control mice partners in terms of the sedentary mice. We saw that those mice were no longer able to run as far. So taken together, we see that the mid mice had at first an initial benefit of exercise, but with the increased higher intensity of exercise, they saw that we actually got an impairment of this endurance training effect. So how does this relate to a number of other studies with respect to these training effects and fuel utilization? And Ray found 2 papers and 2 studies that really speak to what our observations are. The first was done in humans, where a group of individuals were given either a high carbohydrate diet or a high fat diet for 8 days. And they were asked to do cycling tests, 100 kilometer cycling tests, either at day 1 or day 8 on either of these diets. And what was observed was on the high fat diet shown in squares was that there was a clear decrease in the RER shown here. Further, the long distance trial showed no real difference between the high fat diet or the high carbohydrate diet. However, when the individuals looked at power output in the shorter trials of 4 kilometers or 1 kilometer, the high fat diet individuals, when they were on a high fat diet, had lower power output. Again, similar to what we're seeing in our mice. In another study, we found that on a high fat diet, rodents running the same run to exhaustion test also had this impairment that led to, at about 500 meters, they sort of stalled out with respect to their ability to run. And that's quite similar to what we're seeing here in our mid mice. With respect to functional capacity, muscles again exhibited this atrophy that we have seen over time. Force was similarly lower, but when we normalized force to cross-sectional area, there was a normalized strength. Mitochondrial adaptations were also improved in both cases, so PGC1 alpha increased with training, as did indices of the mitochondrial proteins, so the electron transport representatives were also improved. Body composition itself also improved with training, regardless of whether or not these were mid mice or control mice. And shown here is lean body mass being maintained with training and fat mass being reduced with training compared to the sedentary controls. Further, when we looked at glucose handling in the mid mice, we actually saw an improved clearance over time with training. The last feature of this is something that we were not anticipating, but what we have now seen in these mice was that the tibial, the tibia mass and density is actually lower in mid-mice. Training did not really alter this, but it's telling us that something is definitely going on with respect to the bone muscle or muscle bone crosstalk, either with IGF-1 itself or the lower mass of the muscles affecting bone density. So to conclude, what we see is that this reduced levels of IGF-1 really lead to hallmarks of aging with respect to lower muscle mass, lower bone density, potentially. And also, in addition, we see hallmarks of disrupted metabolic homeostasis, including impaired glucose clearance and increased adiposity. We see that exercise training at low intensities improves the running capacity, improves the adiposity and glucose handling. And this shift in fuel utilization towards fats might be the limiting factor with respect to higher intensity exercise, suggesting that in aspects of potentially aging, that high intensity exercise might not be the right approach to take in terms of therapeutics. And with that, I'll thank those who did it. Again, Ray is actually now in Urbana-Champaign. He was assisted by Caitlin Gregorio, a really talented undergrad who's been in the lab and is doing her thesis in the lab. Hanshin and Yorgos have been at his side, helping with a lot of the molecular analysis, and a number of the other members of my lab are also studying different aspects of IGF-1, my collaborators and funding, and I'll stop there. Thanks very much.
Thanks very much, Dr. Barton, Beth. That was really interesting talk. And I've got a couple of questions while others are thinking about their own. So one of the things that first struck me about this is with several anti-aging types of interventions, whether we're talking about caloric restriction or the Snell-Ames dwarf mouse, those species and conditions are associated with lower circulating IGF-1. And so that was given a positive spin in the context of their longevity and health span. But you're kind of advocating for a different spin in your mid-mice. And I'm just wondering if you feel there's a paradox there, or whether you have an explanation for why it might have a different impact.
So one aspect is that, you know, we're tweaking one tissue, right? And that I think most muscle physiologists would agree that higher IGF-1 within muscle is incredibly beneficial, both in an anabolic and metabolic manner. However, the trick or the problem or conundrum is the fact that the rest of the body probably does not like that much IGF-1 over time. And so I think you're right in that how do you, the ideal therapy would be something where we could boost IGF-1 only in muscle and leave the rest of the body with the lower levels. If that answers part of your question.
Yeah, I think that makes total sense. And you and I maybe even talked about this several years ago, because I know measuring IGF-1 protein has been a real pain over time. But now it looks like you folks have got a good ELISA and you're happy with how that's working. So as far as I know, nobody had measured IGF-1 levels in the muscle of caloric restricted mice or rats. So it may be that, you know, circulating IGF-1 is lower, but not necessarily muscle in that context. And so without looking, we don't know, right? So maybe that would also be part of that explanation. And
yeah, I don't know the answer to that, how it would alter that. Yeah.
So we do have some muscles frozen from caloric restricted animals, just FYI, if we ever wanted to look.
Okay.
I had another question. If nobody else wants to interrupt me yet, I'll pause for a moment. Okay, so I'll continue. Oh, there we go. Please go ahead.
Hi, Beth. So I have one comment and one question. The effect on bone would be, I think, is something that we all really should get more into looking at, because it's the obvious effect that you get once you lose the tension of the muscle and the mass of the muscle, you lose the bone. So what you're seeing in the mice, but I think you have a nice model now to be able to look at the crosstalk between muscle and bone, which will be really good.
Yeah, we're quite excited about trying to do that. Yep.
Yeah, so it's a really, I really like this model. It really is like a model of aging, but so it gives you a mouse where you can do all these things without having to age the mice out and spend a lot of money. And the question I have is, have you looked at the neuromuscular junctions, which are also very sensitive to IGF-1 levels?
We have not yet, but I think listening to the talks today, I'm definitely wanting to find a collaborator to look at the NMJ to see how it's how much integrity or whether we've lost it.
Right.
Our functional outputs right now are only done by direct stimulation, so we can't tell whether there's any deficit in neuromuscular junction integrity yet, but we can start to get some indirect measurements for that. Yeah, but it'd be very interesting to see.
Because then that will give us a nice model to be able to look at the effect of intervention on neuromuscular junction, as well as the mechanism of how they disintegrate and you lose them during aging.
Yeah.
Great.
Thank you.
Thank you. I see another hand raised. Georgia,
yeah.
I'll let you.
Paula. Yeah, thank you. Can I go on, Ugo? Sorry.
Please.
Yeah. So I actually, my intervention was in the same line of the GL1. And because I was referring to the graphs when you have exercise animals in the 2 rounds of exercise, and they drop their performance both in the exercise group but also in the sedentary group. So these drops is due to the aging of the animals, or the premature aging, I mean, aging effect, of course, on the performance. Or is the fact that the section damage the muscle and they cannot recover, so they get worse even?
Well, so 2 things. One is that these mice were 28 weeks of age at the end of this study. So that would not be a true aged mouse, but it could be reflective of a potential for aging that's there. The other aspect is that, when they're running, this is whole animal running. So we've not done any damage. We've tried to make sure the animals are on a 0% incline. They're running on the flat surface. So there's no ongoing damage that would occur with the running itself. I think in retrospect, what we should have also included was a running test where they would be at a low intensity throughout the process, throughout the protocol, because my bet would be that they would be able, the mid mice would be able to run longer at a slower speed. And that what we're doing as we're increasing intensity is outstripping their metabolic capacity with these higher intensities. But it's all an open question as to what is happening at that in those last tests.
Did you check if they get more damage after a bout of exercise?
We have not done. You mean in terms of? No, we
fiber damage, or I mean,
we don't see any incidences of that in histologically where we see any rounded or necrotic fibers. The analysis of the muscle histology, however, was 24 to 48 hours after that endurance exercise test, and so we didn't control for the amount of time after that. What would be good is to try to, hone in and actually, look at a defined time to see if we could see that damage. That's a good.
May I join a short comment? Why not to just 1 minute? Just look at the same mice 1 week later. You will find a lot of muscle regeneration.
It's possible.
It is not possible. Yeah. It is sure.
Okay.
I have 20 years of experience on these running mice. They have a lot of damage, background damage that you recognize easily by embryonic myosing a week later. Try and thank me in your manuscript with 5 EGTM references.
Oh, for sure.
Good luck. Sorry
Thank you.
to the chairman.
Yeah, Paula, you had a question?
Yes, a briefly question if I could go, I don't know.
Yes.
We know that IGF-1 and some neurotrophic factors that are in the muscle like BDNF or NT45, they upregulate each other's. You have an idea what could be the influence of those neurotrophic factors in the conjunction of your work with the IGF-1?
We haven't looked, and I think we could go back to the RNA-seq data to see if we can pull out neurotrophic factors. We haven't mined that data set for those factors yet, but that would be a really good key to take a look for neuromuscular junction integrity as well. Yeah, that's a great idea.
I'm working that field also, so you can change some.
Wonderful. I will look for you. Thank you.
Yeah. And thank you for your talk.